I would like to thank Mr. Felix and the organizers for inviting me to this talk. Today I will discuss uncovering modifiers of tau propagation using a human iPSC-4R tauopathy models. This is the outline for my presentation, where I will explain the results that have been discovered. First, I will give a brief introduction. Then I will present our work on development of a normal iPSC-derived 4R tauopathy model, determination of whether tau fibrils induce progressive tau inclusion, identification of normal therapeutic modifiers through CRISPR-I screen, and validation of identified modifiers. Let me start with a quick introduction. Primary tauopathies are classified to 3R, 4R, or 3R and 4R tauopathies by the primary tau protein isoform deposits in the brain. 3R includes pig disease, 4R includes PSP, CBD, AGD, and GGT. 4R and 3R mixed tauopathies includes PART. In healthy neurons, tau is mainly found in exons, where it plays a key role in stabilizing microtubules. In diseased states, tau becomes hyperphosphorylated, misfolds into soluble oligomers, aggregates into insoluble fibrils, and forms neurofibrillary tangles. This process leads to microtubule disassembly in exons and finally results in synapse loss. Tau is encoded by MAPT gene on chromosome 17. The pre-mRNA undergoes alternative splicing of exon 2, 3, and 10, producing 6 isoforms. Alternative splicing of exon 10 produces either 3R tau or 4R tau. Alternative splicing of exons 2 and 3 generates 0N, 1N, and 2N tau. In healthy human brain, equal levels of 3R and 4R isoforms are expressed. Imbalance in 3R, 4R protein ratio can trigger pathological tau aggregation. There are many mutations in MAPT genes that are considered disease-causing. One prevalent mutation, P301S or P301L, is highly associated with FTLD MAPT. Growing evidence suggests the accumulation of misfolded tau is mediated through the spreading of protein seeds from cell to cell, leading to tau propagation and spreading toxicity throughout different brain regions. However, there is no cure for tauopathies, and no drugs are available to prevent or treat them. One major challenge is that drug effects observed in animal models are not always reproduced in human clinical trials. The development of iPSC models to study tauopathies and advanced therapeutics has become urgent. However, iPSC-derived neurons mainly express 3R tau only. The inclusion of exon 10 in these cells is extremely low, making a major challenge to study 4R tauopathies and mutations located in 4R tau. To address this problem, we aim to generate new human iPSC-derived neuronal models that mainly express 4R tau. To increase 4R tau expression, we edited the MAPT locus around exon 10 in an iPSC line, which contains an NGN2 transcription factor in AAVS1 locus. We use CRISPR-Cas9 gene editing with a donor plasmid containing silent mutations in and around exon 10 to promote its inclusion. We further introduced the P301S mutation into this line to study its effects in vitro. We then analyzed the neurons derived from the cell lines by both Western blotting and immunocytochemistry, and confirm a robust increase in 4R tau protein isoform in both homozygous and heterozygous 4R neurons using an ET3 antibody which specifically recognized 4R tau. The 3R tau protein isoform recognized by RD3 was detected only in heterozygous and parental neurons, but total tau protein was observed in all neuronal lines. This observation was further validated with immunocytochemistry. Only 4R neurons were stained with the ET3 antibody. Phosphatase used here is to remove phosphate groups. Further analysis of neurons derived from these lines revealed that 4R lines show significant increases in disease-associated post-translational modifications, including tau hypophosphorylation at multiple residues and acetylation at lysine 174, which is known to block ubiquitination at this site and thereby prevent tau degradation. We then ask if the modified iPSC-derived neurons develop progressive tau inclusions when seeded with tau fibrils. Using MC1 antibody, which recognize a pathological conformation of tau protein, we were unable to detect any endogenous 4R tau inclusions in parental or 4R neurons seeded with K18 fibrils, but we did observe tau inclusions in 4R P31S neurons seeded with K18 fibrils. We also observed an abundance of hypophosphorylated tau and oligomeric tau in the mutant cells. We also observed a progressive accumulation of MC1 positive inclusions in 4R P31S neurons from 1 to 5 weeks post-seeding. To determine whether tau inclusion only depends on K18 fibrils, we seeded neurons with Y-type 0N4R tau fibrils. We also observed MC1 positive tau inclusions, so tau aggregation in these neurons is not limited to seeding with specific fibrils. We then ask if these in vitro lines recapitulate in vivo tau pathologies to study if tau inclusions accumulated in neurons. We differentiate iPSCs into neurons seeded with K18 tau fibrils after 1 week and collected neurons for analysis between weeks 3 and 5. We performed detergent fractionation on unseeded and seeded 4R and 4RP301S neuron lyses to separate soluble and insoluble protein fractions. We observe an increase in total tau and phospho-tau in insoluble fractions of seeded 4RP301S neurons. To track live neurons with or without tau inclusions, we generated a new iPSC line by inserting the holoTag upstream of MFT locals in 4RP31S iPSC line. The holoTag can be fluorescently labeled open binding to a ligand containing a fluorophore, enabling live cell imaging of endogenous tau. As shown here, the red arrows indicate neurons with visible tau inclusion, while the green arrow indicates a resilient neuron. When we measure the spontaneous activity of 4RP31S holoTag neurons expressing the calcium sensor, we observe that neurons with inclusions show a significant reduction in calcium transient spike amplitude compared to neurons lacking inclusions. To investigate potential dysfunction in cellular machinery following seeding, We perform transmission electron microscopic to compare subcellular structures in 4R and 4R-P301S neurons with and without K18 seeding. We observe an accumulation of abnormal vesicle structures with multi-layer membranes exclusively in K18 seeded mutant neurons, both in soma and neuronal processes. Because the accumulation of these structures suggests impaired endolysosomal membrane trafficking. We also observe large tau inclusions in the soma of 4R-P301S neurons when seeded with K18 tau fibrils. We then took advantage of this platform to identify normal tau propagation modifiers through CRISPR-I screening in collaboration with Dr. Martin Kupperman. We engineered 4R-P301S iPSC to stably express that Cas9 fused to a CREB replacer. When the guide RNA binds to its target DNA sequence, that Cas9 CREB complex is recruited, effectively replacing gene transcription. This allows to identify genes essential for specific cellular process or phenotypes. The guide RNA library used was customized based on the tau oligomer screen and tau interactome studies. iPSC were transduced with guide RNA library and then differentiated to neurons, seeded with K18, harvested at 19 days, stained with MC1 antibody, and sorted into either MC1 positive or MC1 negative populations. The frequency of cells expressing each guide RNA was determined by next-generation sequencing. We identified over 500 positive and negative genetic modifiers of tau aggregation. This volcano plot illustrates the genes whose knockdown significantly increases or decreases tau inclusions compared to the cell treated with non-specific control guide RNAs. The top positive modifiers of tau was BPS29. The top negative modifiers are 5 components of UFMylation cascade. Further subcellular classification of the top hits revealed their roles in ER, Golgi, retromer complex, the vesicle trafficking, and the mitochondrial electron transport chain. To assess transcriptional changes induced by each guide RNA, we perform CLOP-seq by transducing a lentiviral library, targeting the top 30 screen hits into 4R-P301S dead Cas9 iPSC cells, followed by differentiation. Analysis of the resulting data yield 12,000 high-quality single-cell transcriptomes after filtering. Here, UBA5 knockdown as an example. You can see the transcriptional changes after UBA5 knockdown. The next step is to validate individual hits identified in the screening. We knock out BPS29 in 4R-P301S cells and observe a 2 to 3-fold increase in tau propagation by immunocytochemistry and flow cytometry, supporting a role for BPS29 in tau spreading. In the MC1 negative group, 5 of 6 components belong to UFMylation cascade. UFM1 is a ubiquitin-like molecule and is conjugated to its target substrates by enzymes. The biological significance of UFMylation remains largely unknown. To validate the CRISPR-I screen, we show that inhibition of UFMylation through shRNA-mediated knockdown of either UBA5 or UFM1 significantly reduces tau propagation as measured by immunocytochemistry and flow cytometry. We next compare the levels of UFMylation in K18's unseeded and seeded 4rp3-1S neurons in soluble and insoluble protein fractions. Immunoblotting with an anti-UFM1 antibody detects free UFM1, reveals a strong reduction of free UFM1 only in the insoluble fraction of seeded neurons, indicating the seeded neurons perhaps have more UFMylation. We also observe that knocking down UBA5 or UFM1 also reduces intracellular total tau levels. To investigate UFMylation's role in tau pathology in vivo, we injected either control shRNA or UBA5 shRNA lentivirus. Both contain GFP into hippocampus of PS19 mice, which express human P301S tau, harvested 4 weeks after injections and stained with GFP and UBA5 antibodies and DAPIT. Injection of UBA5 shRNA lentivirus lower mouse UBA5 by over 50% compared with lenti control. We further use this model to study the effect of UBA5 on tau spreading. In this model, either a control or UBA5 shRNA lentivirus was injected into the contralateral hippocampus, while K18 fibrils were injected into the ipsilateral side. The brain were harvested 4 to 6 weeks post-injections. We observed significantly low tau inclusion by MC1 signal in mice with UBA5 knockdowns, which is consistent with our in vitro results. To further explore the relevance of UFMylation in tau pathology in humans, we perform co-immunostaining of MC1 and UFM1 in postmortem brain of patients with PSP and AD. We observed that higher MC1 positive neurons show low levels of free UFM1, which is consistent with our in vitro results, showing low levels of free UFM1 in tau-seeded iPSC-derived neurons. Our findings support that UFMylation promotes tau pathology and human tauopathies. In summary, we successfully engineered human iPSC-derived neuronal lines to express 4R and 4rp3-1S tau. The 4rp3-1S neurons develop progressive tau inclusion, open seeding with tau fibrils. A CRISPR-I screen targeting genes associated with tau pathology identify over 500 genetic modifiers of seeding-induced tau propagations, including MPS29 and genes involved in UFMylation cascade. The UFMylation cascade is altered in neurofibrillary-tangle-bearing neurons from postmortem human PSP and AD brains, and its inhibition both in vitro and in vivo suppresses seeding-induced tau propagation, highlighting its potential as a therapeutic target for neurodegenerative diseases. I would like to thank Dr. Li Gan for her support and valuable discussions. Celeste, Iris, Hasu, Zeping, and Yansong for their hard work, and Wenjie for helpful discussions. I also thank Martin's group for sharing the reagents, and thank you for your attention.